An introduction to Flavr Savr tomato. A major problem in agriculture is the perishing ability of its products. Various methods have been initiated and developed to increase the shelf life of crops, especially for fruits and vegetables. One such successful technique is delaying the ripening of fruits and vegetables by manipulating the genes through genetic engineering. What is fruit ripening? Fruit ripening is an active process characterized by increased respiration accompanied by a rapid increase in ethylene synthesis. The fruit gets softened as a result of the activity of cell wall degrading enzymes, which makes them more susceptible to being damaged by fungal infections. The phytohormone ethylene is known to trigger the ripening effect. The breakdown of starch to sugars and accumulation of large number of secondary products improves the flavor, taste, and smell of fruits. So what is Flavr Savr tomato? It is a genetically modified tomato which was commercially grown and produced by the California company Calgene and submitted to the US Food and Drug Administration FDA in 1992. On May 18, 1994, the FDA completed its validation of the Flavr Savr tomato and the use, concluding that the tomato is as safe as tomatoes bred by conventional means. On May 21, 1994, the genetically engineered Flavr Savr tomato was introduced. So this is the gene encoding polygalacturonase has been isolated and cloned. First, the DNA was isolated from tomato plant that encodes the enzyme polygalacturonase. Then the polygalacturonase gene was transferred to a vector bacteria and production of cDNA molecules was done. Then the cDNA molecule was introduced into a fresh tomato plant to produce transgenic plant. This was done through the antisense RNA approach in which the polygalacturonase gene encodes a normal or sense mRNA that produces the enzyme polygalacturonase and is actively involved in fruit ripening in the normal plants. Whereas the complementary DNA of polygalacturonase encodes for antisense mRNA, which is complementary to sense mRNA. The hybridization between sense and antisense mRNA render the sense mRNA ineffective. Consequently, no polygalacturonase is produced, hence fruit ripening is delayed. So this is the picture. It is a normal tomato from which the normal sense mRNA codes for polygalacturonase protein and hence it helps in rotting of the tomato due to polygalacturonase. Whereas the inclusion of a synthetic antisense gene produces antisense mRNA, which binds to the sense mRNA, forming complementary mRNA, and hence the production of polygalacturonase is stopped, which helps in keeping the tomato or increasing the shelf life of the tomato for about 3 weeks. So, the advantages through genetic engineering, the ripening process of the tomato was slowed down, thus preventing it from softening while still allowing the tomato to retain its natural color and flavor. Thank you.